start the event, ma'am?
Yes, ma'am. Sure, with pleasure.
Yeah, ma'am. Thank you, ma'am. Good evening, everyone. Warm greetings to all the distinguished participants, faculty members, researchers, and students. On behalf of the Indian Association of Research and Development Professionals, IARDP, in association with SKRGC Academy, it is our pleasure to welcome you to Day 5 of the Multi-Domain 7 Days Faculty Development Program on Interdisciplinary Applications of Artificial Intelligence. Today's session, usage of agentic AI and foundation models to solve biological bottlenecks, highlights the transformative role of advanced AI systems in addressing complex challenges in the biological and life science domains. As AI continues to evolve, its interdisciplinary applications are opening new frontiers in research, innovation, and problem solving. We are honored to have Dr. G. Brindha ma'am, Assistant Professor, Department of Biotechnology, Hindusthan College of Arts and Science, Coimbatore, Tamil Nadu. As today's resource person, her expertise and insights will provide valuable perspectives on how agentic AI and foundation models can accelerate biological research and overcome critical bottlenecks. We encourage all participants to engage actively, reflect deeply, and make the most of this enriching learning experience. Welcome, ma'am. Welcome once again. Wishing you an insightful and inspiring session. Thank you all and welcome once again, ma'am.
Thank you, ma'am. Thank you so much for welcoming. So, very good evening to everyone present here. I would first of all thank Indian Association of Research and Development Professionals (IARDP), which is a unit of SK Research Group of Companies in association with SKRGC Academy, for inviting me to be as a resource person in the multi-domain 7 days faculty development program on the specific topic of interdisciplinary applications of artificial intelligence. I once again thank you all for providing me this opportunity. Ma'am, can I start the session?
Sure, ma'am.
Yeah. Ma'am, just let me know if the screen is visible.
Yes, ma'am. Is it visible, ma'am?
Yeah, thank you so much. So, as like we have discussed, we have today my topic is mainly about solving a biological bottlenecks with agentic AI and foundation models. So, coming to this, you all know what is actually biology is all about. So, biological research today faces a lot of paradox. On one hand, AI can design proteins, drugs, and even gene edits in just few seconds or minutes. So, on the other hand, validating them in the lab takes more months or years. So that is there the gap between a computational experimental reality. This is what you call it as biological bottleneck. So what's the gap between your biology lab, what we are doing it, and how is AI taking it out? So today's lecture will tell you how this agentic AI combined with the foundation modern research is transforming your biology from slow manual science into semi-autonomous discovery. So this is what is all going to discuss about. So coming to the slide part and the explanation part of biological, which you call it as DBTL, which is nothing but design, build, test, learn, a loop. So this is all about the biological information where we discover the drug which we take in. Normally it can be any drug, a very small paracetamol or any antibiotic or anything which is there. It's just drug discovery, your protein engineering, and especially your synthetic biology is like which you discover something, and or gene editing which operate through a fundamental framework which you call it as design, build, test, learn, loop. So this is what is all about happening in and around whatever the main sector may be, but this is what we're facing about. So this is a literative cycle which defines how biological ideas are translated into experimentally validated outcomes. So here, coming to the design phase, we researchers or you people will use computational tools to create this hypothesis, such as designing novel protein sequences, identifying the drug what is required, and planning them. But now your modern AI Your modern stimulations and all those large biological databases have enabled this to an extremely fast, and it has made the world a wide vast. So advanced AI models can rapidly generate thousands of candid in the sense of very important molecules, which is very useful. For example, if you take breast cancer or any other cancer which has been a risk of on the traditional area, but now what happens? Within fraction of seconds, using this AI, people are able to find out at least what the patient is suffering from, whether they are suffering from normal cell or it's a cancer or any other infectious disease. So here, the build phase will always involve you covering the converting a computational design pattern into physical, entitled one, and this always includes whatever test you go with DNA isolation. And coming to biology part, it's all about the protein expression into the host system and cell engineering system. But the test phase will tell you whether your built biological system performs as intended. And this is what it tells you about the functional phase to measure your biological activity, toxicity, and safety testing. And the final stage is whatever you do, whether it's AI, whether it's biology, the final part, what we do is always for the learning part of it. So when you design it, you build it, you validate it, which is nothing but the test part, and finally comes the learning aspect, where it involves analyzing your experiment data, usefully the inciting sites into the iteration. Ideally, the feedback should continuously improve your model predictions. But how do you do that? It all comes through a learning part, which limits your efficiency of the overall entire cycle which has been adopted to us. So the core, coming to this biological bottleneck word, it always lies in a mismatch between the speed of computation and the space of experiment. So when you design, you build, you test, it takes weeks to months. But when you talk about a clear real world example of this bottleneck, it was, see, a very simple example I'll give you, which has happened during a COVID-19, which is a pandemic, this is you all know very well. And these AI based computational tools are able to rapidly design and predict how this COVID was, what role it was playing with this the entire universe. How do you say this? How people all died because of this? It's only through AI we were able to predict it. Rather than being a biologist, we were not able to find out that it was a virus or this the disease causing it. So the key insight from this is biology today is not limited by creativity or computational intelligence, but the spread, scalability, and automation of experimental validation. So there are about foundation models or in biology, or you compare, give a compare between the AI method, which you call the brain of it. The foundation model represents a transformative class of artificial intelligence system that function as the cognitive core of modern biological AI. So these models are large-scale neural networks trained on to an unlabeled biological data. So what do? You find out something. Something can be done from your blood or any sample from your body. You find it out, you make a model of it, and then again you have to go again with your any of your AI model. For example, you go with ChatGPT, which will tell you what type of schematic happening is happening in a human body, reading the massive text of your corpora. So one of the most popular example will be your Evo, which is the trained one. It is a genome which will tell you about how this happens here. This will be compared with any other person in this world, but how it can be done only through AI. All these are softwares, that is Evo, DNA BRAT, Protein MPNMM. All these are AI softwares where you compare it with your biological aspects which we isolate and do in the lab. So what happens here? You have your genome scale evolutionary process. So a particular important application is the identification of disease causing organisms. Yes, see, when we cough or when we have fever or anything it can be, will not be aware of it. So immediately you'll say, okay, fine, I drank water outside, I went in the cold climate, that's why I'm getting all this. But how does it cause? It is all because of the microorganisms in and around you. But what you do, you go to the Google, you go to the AI method, that will clearly tell you. Before all, we have to go to the doctor to know what is happening in our body. But now the technology has changed literally. You just sip out your symptoms in the AI way. It will clearly pour it out to you what is happening in your body, what is the medicine required for you, what diet you should take and follow. So that is how all these claims are. So when you talk about these prot agents, it is literally a very simple when it comes to a predictive system, what is happening in you. So it is all about like what types explain to you about the labor intensive process in biological research. For example, if you take a traditional mechanism which is happening around, you have to identify that. If you take the old method, see, before all traditional food we never respected it, it was years back. But now what we do, wherever you get your traditional food, you're ready enough to pay them with so much of amount. You're ready to spend expense on them, so that you want to make yourself healthy. But before it was not that way. When they say traditional, no, the kids were not ready to take that. But now the scenario has literally changed, because when you take the fast food or something, some issues are happening in your body. So how does this been predicted? That's what you call it as autonomous discovery. Here, autonomous means self-replicating, which is happening inside your body, and you have to discover it at the earliest point. So, what is this prot agents? They are very simple. They are autonomous protein engineering happening into your body. This will tell you what agents are affecting it, what protein engineering will happen to you, what mechanism is happening inside your body. So for this, again, you have to go ahead with your foundation model, such as protein MPNN, which is a protein detector coming to the biological part. It will tell you what is being affected for you, what food you have taken it, because of that, which part, whether your stomach or liver or kidney, or which part is affected. All these will tell you about in an experimental feedback, reducing your human intervention and building you a good life. So in a real world application, when you say that this has got a very good potential application, whereas in industrial wise it is used for therapeutic development. Here the therapeutic development means developing of new tablets or medicines or anything which is very crucial to be brought out. Whereas when you talk about, you all know about vaccines. Yes, of course, yeah. The one, even for COVID, we have COVID vaccines, or measles, mumps, chickenpox, anything you develop vaccine. But proteagens can assist and optimize it. What is the mechanism happening there? And coming to the next of the CRISPR part, yes, a CRISPR part is a very sincere one, because this now the recent technology is entirely dependent on this CRISPR gene, which is the gene in your body. Each and every human being will have this thing, where this gene is responsible for whatever difference mechanism is happening inside us. Whatever it can be, yes, we are solely responsible for it. It is based upon the, see, like Chat GPT, you have a software called as CRISPR GPT that will tell you your what is required, what is your body doing, which gene you have to take it out, what is the mechanism happening inside your body. If one gene is there, okay, this gene is responsible to produce hair, this gene is responsible to produce sweat, this gene is responsible to produce your color. So you take out that gene, you give it in a software called as CRISPR GT, that will give you your entire body, why this male is this color, why this female is this color, or else why this color has been changed for them, what is lacking in them. This is how your technology goes, which you call it as optimal gene editing strategy, which is all about your specific mutation or therapeutic goal. So, for example, if you take out your prostatic cancer, any cancer, you give out the gene, that will tell you, okay, why you have got this cancer, what was the food you are lacking in, what is the gene mechanism, or your father, mother, your grandfather, anyone who from whose generation it has been come. All these combined together will tell you what are this. But the only thing, the drawback here will be when you talk about AI, you will not be able to say that this is the exact reason for this. It can give you ideas. Again, I'm telling you, it can give you ideas, it can give you, it can give you everything. But we will not be able to sort out this. So where comes, you have to come in with your biology part. So coming to the outlook challenges when it's happening, of course, yeah, you should have a data if you're thing. And apart from that, you have other concept which is happening, which is nothing but permanently connected to a laboratory instruments, and self-improving your biological discovery system, data quality, black box nature of advanced AI models are very important, which has been recently used everywhere. And you talk about your ethical. So all doing all this is fine. You go with AI, you do this, but where is ethics going on? Can you do anything without ethics? Of course, no. You're going to take a blood sample, you should have an ethical letter with you. You should get a sign from the patient. This AI can't do, right? So in that way, you have to predict the software system, or predict or develop something which is related to your AI, which tells you about your concerns of what is happening around you. So what is all about a self-driving laboratories? Yes, it tells you about what all the tools you can be involved in your molecular biology basis or genetics or whatever mechanism happening related to biology. It will tell you about your slow manual experimentation, faster drug development, smarter gene design, reduced cost and experimental failure, and a very good data which is very much required for you. But how does this again happen? You use your black box nature of advanced AI models. You can use your foundation models and agentic system, which will make a highly accurate and precise your predictions but provide limited, very, very, very limited insight into the how decisions were reached. And for example, you must have all known about your biomedical, clinical laboratories, or any labs in and around you, any testing labs, any blood testing lab or scan lab, anything which is in and around you, you can lack their interpretability will be very problematic. So that time again, your AI is the only tool like your PyMod or Uniprot or anything like that, which will tell you about what is happening there, how what is the mechanism happening inside your body, what is the reason behind it, how do you predict it related to drug safety, gene editing, or disease diagnosis. And then you bring out the data quality. Your biological data is very, very, very incomplete. As a person, you will not be able to stick out your biological incompatibility. So that time you have to bring out your tools of all your designs. So when you talk about in vitro, in vivo, very simple. You must have known about IVF. Yes, doctors go with IVF when a person is not able to get a baby. So that is literally in vitro. You take your persons, both father and mother, you develop in a lab, you give them a baby. Whereas in vivo, you take it happen inside your human body. But now again, as scientific as developed, as computer stream has become the most widely used, being adapted the entire universe, you go with the laboratory. Now wherever you see that laparoscopic, robotic laparoscopic surgery, of course, yeah, and it has been the successful one. Why? It will assist just what you say. But as a person, when we design an experiment or when we do some surgeries, there will be mistakes where we go ahead with another process. So in that case, of course, your build, test, learn cycle comes out with your major aspects and everything which is happening around you. A very simple laboratory surgery which has been research is being taken on will be your SAP and DMX platforms. A very, very, very important thing. So all these, you come into the case study, your proto-agents, that is your automatic design, testing, and refinement means it's like you bring in some changes of what you're doing. It doesn't mean that you have to go whatever your universe says. You use your lab agents and you make that refinement, you conclude yourself, that's what you call it as refinement. And apart from that, you have your CRISPR GPT, as I told you, like your chat GPT. You have a gene GPT where you give any gene, because in your body you have around 28,000 to 30,000 genes in your body, and each gene is responsible to produce a protein for you. If that gene is fine, then you're a perfect human being with no disease in you. That's where we are all making mistake there. And as a person, like DNA, your per DNA and my DNA will be 99.9% will be the same. Only that 0.1% will always vary. So coming to this transition, of course, yeah, you all know that it's a very, very difficult one. So biology is entering a transformative era where intelligence is no longer confined to a human alone. Foundation models act as the brain, the learning grammar of life from cosmological data, while the agentic AI, which I told you, it is just your accompanying partner, or simply you can say a scientific partner capable of planning, executing, and learning something from the experiments which you're designing. So when you talk about the difference between this bottleneck or this foundation model or agentic AI, it's very simple to say that this foundation model or the agentic AI will always be the hectic part of it, because you design something but you deliver something. That's where everything happens around. So by integrating this prediction with action, your biology moves. It will be a very slow manual experimentation, autonomous self-improving discovery system. And this faster drug and vaccine development, it's a smarter protein and gene design. It will reduce your cost, and it will always have an experimental success. But when you talk about your process demand or declaration, it's like your transference and interpretable AI models, and a very, very, very strong ethical and biosafety regulators where nothing can be done with anything else. And of course, the future of biology is not AI replacing scientists, but AI collaborating with scientists. I want to make this sentence very clear. Never say that whether this biologist will do all the work or AI will do. No, it's like it's always a collaboration work. Now, a computer science hands hold with a biology, because biology can give the data, but computer science are the main top notch people who bring in out what is the final result or what is the current scenario happening there, which will accelerate your life saving discoveries. So, finally, I concluded saying that AI can be not only a tool, it can be a scientific partner where multiple personalities can be bringing out and significant reduction, because as a biological person we'll know what to do, but as a user of a tool, You are the people which will tell you what is the mechanism happening around you, what will be the current scenario, what will be the entire data they are going to work on it. So it's all about you people, which tells you about what is the mechanism happening around. So it's always a joint hold hand, as I said earlier. It's a joint hold hand between the AI with with the stream, along with the people of biology. So, bottleneck or foundation or any disease or any prediction, of course, yeah, it's the role of a biology people who will discover it, who will work on it. But at last, they will be the part who will be able to sit out with the people of AI tools. With, see, for example, if you take protein, you'll have a protein database. If you have a DNA, you have a DNA database. But it's with the AI which will combine and tell you whether this comes under this category. That's where we are all lacking about, and that's what we have to work on all about. So I hope you all have an idea about what is bottleneck, what are the foundation models happening around, and what is the scenario connected between a biotechnology and a multiple domain stream with AI. So this is what we finally bring on it. We bring on with a combination of what's happening around, and we'll tell you what this tool is used for. Again, as it has an FDP, we can bring out saying that AI plays a major role, but without our solution, AI can't do anything. It will just give out whatever we ask you. But we are the people who have to work on it, we have to solve it, and put forward the entire success of it. So once again, I thank you, SKRGC Academy, for providing this great opportunity to deliver you all. Any questions, please let me know.
Participants, if you have any queries, you may ask.
Huh. Well, if there are no queries, then sure we can wind up the session.
Ma'am, what is that biosafety concern, ma'am?
Sir, when you talk about biosafety concern, like we are just working on with certain aspects, right? So, you, for example, we do some biological process. We go to the doctor, you do a test. Any test you do, you take about your samples. So, for that, we should have an approval of ethics or anything like that, where you should be very clear of what is happening to you. That's the main part where we should be able to explain. Though the results are coming through AI, anything like that, it refers to the measures and practices which is designed to protect your human, animals, and the environment from all your biological agents like microbes, the toxins which has been delivered to you, as well as through you whatever the food you take now through genetically modified. There are so much of products have come which has been literally changed through the gene modification. So especially for you when you take or if you go to laboratory, biosafety concern, if they take your blood sample or something, like it's their duty to put it into the dustbin in front of you, so that you should be very cautious that they should not use the same needle for another person. So it's the concern of first thing is risk of infection, next is accidental release of pathogen into the environment, and properly handling of all your equipment. For example, if you're going to handle any like COVID virus, see, it created a boom to the entire universe because of the handling of the misuse. It started spreading around where we're not able to control it up before the vaccine development. So the biosafety concern is very simple for you to say that it is like it measures and practices and tells you how you protect your human or animals or anything which is related to the environment.
Yeah,
Shall,
thank you, ma'am.
Thank you, sir.
shall we conclude the session, ma'am?
Yes, sir, sure, with pleasure.
Thank you, Brindha, ma'am, for your valuable and wonderful session, ma'am. I sincerely thank all the participants and the coordinating team for their efforts. We shall meet tomorrow in the next session. Thank you all.
Thank you,
Sir,
thank you.
tomorrow is the last day, na, sir?
Sir, sorry, sir.
Tomorrow is the last day, na?
Yes, sir, of course, sir. Sir, sorry, sir. I think there is a session called 25 also, sir. 25 in January.
Okay, then. Okay, then.
Yes, sir.
Thank you.
Yes. Okay, sir. Thank you, sir.